Okay, well, welcome to Chapter 7, Outline. Let's talk about cellular respiration. Again, we have this redox reaction that we talked about when we talked about photosynthesis. Again, we have an oxidation and a reduction, hence the redox, right? Oh, I guess I should put it there, redox. So remember, oxidation, we're going from a larger to a smaller, so we're losing, loss. And a reduction, we're going from less, we're getting more, so we're adding. And that we're specifically talking about electrons. So I just want to make sure in this process we understand this is really the opposite of photosynthesis. So this is cellular respiration. We're taking glucose and oxygen and turning it into carbon dioxide, water, and this energy we're going to store in terms of ATP. So this is the food that you eat plus the air that you breathe, air in, we'll say. This is the air you breathe out. This is water. Remember, water is a necessary solvent. We have it in our blood. We need it for our cells. And then we have energy, which we need to do work. If you remember, photosynthesis is the exact opposite, right? Plants take in carbon dioxide and water, and in this case sunlight, and turn it into sugar and release oxygen. So this is just the opposite of photosynthesis. These are the 4 phases of cellular respiration. I'm going to draw a line right here. This does not require oxygen. So sometimes you'll hear this called anaerobic. So an meaning no, aerobic meaning oxygen. These steps you need oxygen. So you'll hear this called aerobic. So let's look at, this is kind of a cartoony picture that you can see. Here's glucose that's being brought in by a carrier protein. Now we have our glucose, and we're going to carry out our 4 steps. Here's 1, 2, 3, and 4. And we're going to take this glucose and we're going to make tons of ATP. And so this is our goal, is to make these ATPs. All right, so we're going to start with glycolysis first. So glycolysis, you're taking a 6 carbon, you're using 2 ATP to split this 6 carbon into a 3 carbon and a 3 carbon, but there's 2 of them. This should look familiar, G3P. What did we do for photosynthesis? Photosynthesis, we took 2 G3Ps and we made glucose. Well, in this case, we're taking a 6 carbon and making it into 2 3 carbon G3Ps. We have to put in 2 ATPs. Sometimes you'll see people call this the energy investment. Basically, you have to put energy in order to make energy later. So here's our 2 3 carbons, right? We're going to put in NAD plus, and we're going to get out NADH. This looks very similar. What was it in photosynthesis? NADPH, right? Now it's not the phosphate part, it's just the NADH. But what happens here? If we have a positive, that means it was slightly more positive than negative, and now we have a neutral molecule and a hydrogen. What are we stripping from the G3P to make NADH? We're stripping hydrogens and electrons. So we're taking NAD, we're adding hydrogens and electrons and producing NADH. There's 1 on each side. So we're transforming this G3P, and the whole time we're transforming it, we're stripping electrons or energy or hydrogen off of it. So this first step, we stripped off hydrogens and electrons. This next step, we have ADP and we made ATP. So what did we have to strip off of it? Energy. So from BPG to 3PG, we've now stolen energy from that molecule. Now some water comes off, and we break these all the way down to something called pyruvate. Pyruvate is the key step. Pyruvate is what you need in order to enter into the second phase or cycle of cellular respiration. One of the things you need to see on the side, remember we used 2 ATPs to get this started. Now we made 2 ATPs, and then we made 2 more ATPs. So we made 4. We used 2 to start. How many does that leave us with? 2. So you might hear net gain versus total made. So what question could I ask that might be a little confusing? How many total ATPs are made during glycolysis? Total made is 4. If I said give me the net number of ATPs made after glycolysis, the net amount is 2. Some people like to think of this as a paycheck. So you make $4, the government takes $2, your net income is $2. So another way to look at it that might be helpful is to do the input and output chart. So for glycolysis, you have to put in glucose, you have to put in NAD+. What do you get out? You get out 2 pyruvates, you get out 2 NADHs, and you get out a net 2 ATPs. Okay, so this is, they want to make sure you understand inside the mitochondria. So I could go back here. We just finished this section right here. To get into the mitochondria, we're now talking about the preparatory reaction. So the preparatory reaction is going to take these pyruvates and it's going to modify them by adding this CoA, and it's going to strip off some carbon dioxide. So we've gone, remember pyruvate is a 3 carbon. So we're going to break a 3 carbon down to a 2 carbon plus a 1 carbon that's in carbon dioxide. So carbon dioxide is going to be given off. This is waste for us, right? We breathe out carbon dioxide. In this process, we're also, when we change the 3 carbon to the 2 carbon, we're also going to strip some electrons and some hydrogens off. So Krebs cycle is the next cycle. Sometimes you'll hear it called the citric acid cycle, or CAC. Either one is fine with me. You should know both. And I'm going to show you the cycle first, and then we'll come back and talk about inputs and outputs. Okay, so in this process, here's that acetyl-CoA. Remember we had a 3 carbon pyruvate, let me get it out, and then we broke that down into a 2 carbon acetyl or acetyl-CoA, and then our carbon dioxide that we are going to exhale eventually. All right, so this acetyl-CoA goes into the Krebs cycle, and basically this CoA is a tag that allows that molecule to enter the Krebs cycle. So without the CoA, you cannot get that group or that molecule into the Krebs cycle. So this is 2 carbons, right? These 2 carbons are added to these 4 carbons, and now we have a 6 carbon molecule. As it moves through the cycle, if we're going from NAD plus to NADPH, what are we stripping off? We're stripping off electrons and hydrogens. Now we take this 6 molecule, we strip off 1 carbon, so now we have 5 carbons. And then we go from NAD to NADH, we strip off more hydrogens, more electrons, and then we pull off another carbon. We're also going to make some ATP, which means what are we stripping off of this molecule? We're stripping off energy because we have to make ATP. We're down to 4 carbons. We're also going to strip off more hydrogens and more electrons in a new molecule we haven't seen before, which is called FAD. FAD goes in and it holds on to electrons and hydrogens and carries them to another destination that I'll show you in just a second. So basically we're modifying this 6 carbon, we're turning it into a 4 carbon, and we're stealing hydrogens and electrons, and we're stealing energy from that molecule along the way. Now we have to take that 4 carbon and turn it into our starting 4 carbon, oxoalloacetate, and we're also going to steal a few more hydrogens and a few more electrons. So if you look at this overall picture, what are you doing? You're adding 2 carbons to a 4 carbon, creating a 6 carbon, and then the whole time it goes through this reaction, what are you stripping off? Electrons, hydrogens, carbon dioxide's energy to make ATP. More electrons, more hydrogens are being stripped off of those molecules. So basically you're modifying this 6 carbon, stealing electrons, hydrogens, energies, releasing carbon dioxide, and all of those are eventually going to go and do work for us someplace else in the body, or in this case someplace else in the mitochondria. So go back to your inputs and outputs. Remember, we had from 1 glucose we created 2 pyruvates. From 2 pyruvates we created 2 acetyl-CoAs. So we have 2 acetyl-CoAs go in, 6 NAD pluses, 2 FADs, 2 ADPs plus Ps, and what do we get out? We get out 4 carbon dioxides, 6 NADHs, 2 FADHs, and 2 ATPs. 1, 2, 3, 1, 1. 1, 2. 2 carbon dioxides, excuse me, 3 NADH, 1 ATP, 1 FADH2. But this was for just 1 acetyl-CoA. So take all of these and multiply by 2. We should have our 4, 6, 2, 2. 4, 6, 2, 2. Okay, so we are now at this point. Well, I lied. I'm sorry. We're here at this point. So we're after the Krebs cycle. We created NADHs during glycolysis. We created NADHs during the preparatory reaction. We created NADHs and FADH2s in the Krebs cycle. All of these are going to get funneled into this electron transport chain, and there's that word again, chemiosmosis. We're going to use these electrons and hydrogens in order to generate ATP. So this is a picture of the electron transport chain. What's going to happen is NADHs are going to get dropped off. They're going to split. We're going to have the NAD. So if we go from NAD to NADH to NAD, what are we dropping off? We have to be dropping off electrons and hydrogens. So let's follow the electrons first, and then we'll come back and follow the hydrogens. So the electron travels through these complexes and comes out. This is all the electron transport chain. We've seen this before, right? But this is in cellular respiration, mitochondria. We saw it when we talked about photosynthesis. We saw this in the thylakoid membrane, right? Okay, so the electron transports through these proteins all the way to the end, and then the electrons combine with oxygen and hydrogens, and they're going to form water. This should look very familiar. What did we do in photosynthesis? Photosynthesis, we split this into electrons, hydrogen, and oxygen. What are we doing with the electrons in this case? The electrons come around, they combine with oxygen, they combine with hydrogen protons, and they turn into water. So you could say the electrons in glucose, the electrons that we stripped from glucose, where do they end up? They end up in water. All right, follow the electron as it reaches through each one of these protein complexes. It takes a hydrogen and pumps it to the other side. Do you see how as the electron passes, it causes a hydrogen to be pumped to the outside? And this is the cristae, or this is the matrix. We're talking about the folds within the mitochondria. So if we want to go back, we're talking about these folds within the mitochondria. All right, so the electron travels through. Hydrogens get pumped as the electron moves through. These hydrogens come back around to this side. They travel through the ATP synthase, and they generate ATP from ADP and P. This is just like photosynthesis. As the hydrogens move through the ATP synthase, it generates enough energy to take ADP and P and turn it into ATP. So what is this called? This is called chemiosmosis again. So chemiosmosis occurs in both photosynthesis and cellular respiration. Where did these hydrogens come from? These hydrogens came from glucose. Hard to write glucose for some reason. I keep missing the, there we go. So these hydrogens came from glucose. Where do the hydrogens end up? The hydrogens end up as water. We can follow that pathway. So the hydrogen was in NADH. It gets dropped off, pumped across, comes back around, goes through the ATP synthase, creates ATP, but you see that the hydrogen isn't being used in the ATP. It just helps create the energy. Then this hydrogen comes over here with this water and we generate, this hydrogen comes over with this oxygen and we generate water. All right, so what about FADH2? So here's the FADH2. If FADH is converted into FAD, what else is it dropping off? It's also dropping off hydrogens and electrons. They enter this complex right here and then move through this complex. The electrons still move through. Where do these hydrogens go? Well, these hydrogens hang out inside. They're the ones that get pumped through the molecules. They're also the same ones that get combined with oxygen to form water. They're also the same hydrogens that come back around and help generate ATP. So this is the overall product or pathway, if you want to call it, of cellular respiration. So this right here is glycolysis. We take glucose and we turn it into 2 pyruvates and we get a net 2 ATPs. Then we do the preparatory reaction where we take the pyruvate and we convert it into the acetyl-CoA. We have some carbon dioxide come off and we generate NADH. We also generated some NADH with glycolysis. Then when we carry out Krebs, we make 2 ATP, we make 6 NADHs, 2 FADHs, and we have 4 carbon dioxides come off. So there's 4 ATPs over here. When we take all of these hydrogens and electrons and push them through the electron transport chain, we end up with many, many more ATP, anywhere between 32 and 34, depending on how well those protein complexes are working. And again, this is all based on enzymes. There's lots of enzymes responsible for cellular respiration, so every individual is going to be a little bit different. We also take the oxygen and we convert it into water with the help of these hydrogens, right? So these hydrogens and the electrons all eventually combine to form water. So this should make sense because we're going from glucose and we're taking in oxygen. Why do we need oxygen? If we don't have oxygen at the end of electron transport, we have nowhere for these electrons to go and the whole system will shut down. So you have to have water to be the electron acceptor, is what they call it, and form water. So glucose and oxygen come in, and what do we get off? We get off CO2, water, and ATP. So we can see the water here, all the ATP here, and then where's this CO2 come from? All of this CO2 from the preparatory reaction in the Krebs cycle, we exhale. So I know this seems really complicated. I would just watch the video again and go through every pathway and make sure you understand where these items are coming from, where they're going to, and where they originally came from. And when you get to the Unit 2 questions, there are some, in the Unit 2 assignment, there are some questions that will help kind of guide you with the type of knowledge you need to be able to have to answer test questions. So I hope that this helped. I know there's a few more things here. Let's just look at fermentation briefly. Fermentation is if there's no oxygen. So no oxygen, you stop after glycolysis. Remember the picture where we said you have step 1, 2, 3, and 4. All of these happen in the mitochondria. This is the division line. So this is no oxygen. You have to have oxygen for step 2, 3, and 4. So if you have no oxygen, we're not going to do glycolysis, or if you don't have a sufficient amount of oxygen. So what's going to happen is the glucose is going to break down still into pyruvate. You're only going to get a few ATPs. If you remember, you only get 2 net from glycolysis. But this pyruvate can be broken down into lactate or into alcohol depending on the organism. So alcohol if we're talking about yeast, and lactate if we're talking about kind of animal cells. Again, if you're not breathing in enough oxygen and you don't make enough ATP, your body just does glycolysis to get by. You just make enough ATP to get by, and in the process you're creating something called lactate. Oops. Have you heard of lactic acid buildup? If you're not getting enough oxygen, the lactate or lactic acid will build up, and that's what causes your muscles to be sore. This is also the way in which we make alcohols, right? Fermentation process, beer, alcohol, wine. I also wanted to show you that there are several pathways in which food can enter into cellular respiration. So it's not that you have to have food enter just in step 1, 2, 3, 4. It doesn't always have to work this pathway. You can see here, you can take the proteins from the meat, like the pepperoni and the sausage, and the proteins can enter at any one of these levels within the cycle. So you can have the protein just come in here to the acetyl-CoA and just carry out this second half of cellular respiration. So fats and carbohydrates and proteins can all enter cellular respiration at different levels. The last thing I want to mention is we should see the overall picture of how this works together. So you should be able to see photosynthesis happens in chloroplasts. We take the sun, and from the sun we make carbohydrate. We take in, if you're a plant, you take in carbon dioxide and water with the sun and turn it into carbohydrate and oxygen. Well, it's a really great thing because animals need to take the carbohydrate and the oxygen, and they're going to turn it into ATP, energy to do work. The release, or the byproduct, is carbon dioxide and water, and what a great thing, because that's exactly what the plants need to make more food. But one of the things that I want to point out from this picture is it's not just about the plant and animal. We consume the plant food, but I want to make sure you realize that they don't make, I want you to realize that they don't make food just for us. So one of the things to realize is, what is the plant doing? So this is a plant. Plant has both of these. Plants have mitochondria, plants have chloroplasts. So what does the plant do? The plants, this is why they call them autotrophs. The plant can use the sun, use the carbon dioxide and water, it can make its own food, it can use that food to make energy. And then what can the plant do with that energy? The plant can grow. So plants don't make glucose for us to go and pick out of the ground and eat. The plants make the glucose for themselves so that they can grow. We just happen to harvest the plant and utilize the glucose, and we turn it into ATP, which we can use for movement. All right, well, I hope this helped you understand cellular respiration in Chapter 7 a little bit better. I'll see you guys next chapter. Bye.